Hey guys! The project is on plant and ozone pollution. So first we're going to talk about photosynthesis, which I'm pretty sure we all know. So we have the sun, and then the plant. And so the plants get the sun's rays, and then use that to make food, and then they intake carbon dioxide to create oxygen, and that goes through the stomata, which are the little pores. And then ozone. There are two types of the good ozone and the bad ozone. And the good ozone exists in our atmosphere. And then the bad kind is created ground level. We have cars and factories. And they create smog and other air pollutants, and this smog combines with the sun and it creates ozone. So my project is about ozone injury, and it's basically injury to the plants because of ozone. So we have plants, and like I said, during gas exchange, they intake carbon dioxide, but sometimes ozone gets in too. And here are some symptoms of ozone injury. First one's called chlorosis. And it's pretty much when, well, you see this, like, plants are supposed to be green and really nice, vibrant green, but with chlorosis, they get a chlorophyll deficiency, and so they get yellowed or pale, and they can eventually die from this. Another symptom is necrosis, which is like a decaying of the tissue. On plants, it means that spots will darken or get weaker, and then the plant will get more susceptible to other diseases. Other symptoms include flecks, kind of like little tan spots, stipples, kind of like small darker spots on the plant, and sometimes plants may experience reddening, but yeah. The ozone injury usually occur on the upper leaf surface of the plant. And now we have my experiment, which was to observe the effects of plant pollution on photosynthesis and chlorophyll production. So my plants I used are tomato and ivy, and then I had to make greenhouses. And here are my plants in the pot. And I took a crate from my garage, and I got plastic drop cloth from a hardware store like Home Depot. And I had wiring to hold it up. And then from my teacher I got an ozone machine, and I just put it underneath the drop cloth and sealed it with one of the crates. And then in order to measure carbon dioxide intake, I took a probe which is connected to like a vernier lab view thing. And then the next part was thin layer chromatography, and basically like I took these, I crushed them in a mortar and pestle with alcohol, and then put them in a test tube, and then made slides with silica gel and put them in ethanol and watched the colors. As for my results, my plants did show visible signs of ozone injury, and carbon dioxide intake for the plants that were exposed was lower than that of the regular ones. And during the thin layer chromatography, the exposed plants had very different chlorophyll results than the regular plants, but I'm not sure if that is because of lab technique or if that's how it naturally is. Anyways, that's my project, and thank you.